Hi, Assalamualaikum everyone. Today our group is going to make a presentation for our BIO464 field trip. First of all, I'm going to introduce our group members. So we have Elyea, AMIRA, HAWA, NURIN and AMNI. I'm Elyea and I'm going to start this presentation by telling our journey during this field trip. So on 7th of January this year, our class went to Paya Indah Wetland as our first field trip destination. We leave our campus at around 8 a.m. and arrive at Paya Indah Wetland at 9:50 a.m. We are actually supposed to arrive faster, but we kinda got lost on our journey here. When we first arrived, we were welcomed by the rangers and they gave us a briefing about the place. Oh, and we were also greeted by the screaming peacock. This was actually my first time hearing their voices and it's quite fascinating to be honest. So, during the briefing, we found out that Paya Indah is actually a conservation place for migrating species and wild animals. The first activity that we did here after the briefing was feeding the species of hippo, or their scientific name Hippopotamus amphibius. Based on what the rangers said, the hippos we met in that place is called Cendana and it is 25 years old. It also has a daughter named Indah, which is 1 year 7 months old. Fun fact about their hippos, they were actually gifted by Botswana government in 2001. So they are not really Malaysia species and actually imported from other country. Now I'm gonna tell some other facts about hippos that I learned from the rangers. So, hippos are herbivore, which means they only eat plants to survive. The ranger said their hippo only eat special grass and fruits and they need to be fed every day. Hippos are also very territorial. They don't let other species invade their habitats, so they will mark their territory with poop. They will wax and spin their tail around, flinging the poop far and wide to cover their territory. Hippos' jaws are very strong. They can even beat crocodiles, so you have to be careful when feeding them. Don't let your hands too near with their mouth. Another interesting fact about hippos, they secrete hipposudoric acid, which is a red pigment found in the skin secretion of the hippo. Although the secretions are often known as blood sweat, they are neither blood nor sweat. The acids they secrete functions both as a natural sunscreen and as an antimicrobial agent for hippos. So, next time when you see hippos, don't panic if the hippos have red liquid on their body because it is not blood and it actually to protect themselves.
Ya, dia makan. Jatuh pisangnya.
After feeding the hippos, we move on to our next activity, feeding the crocodiles. To get to the lake full with crocodiles, we have to take like a 10 minutes walk to get to the crocodile pond. We don't actually get to feed the crocodile here by ourselves like what we did with the hippos because it's dangerous of course and only the professionals can do that. But here are some of the videos of the rangers feeding the crocodiles. Now I will tell you guys some facts about crocodiles based on what the rangers said. So in this pond, there are about 65 or more crocodiles and these massive fierce looking crocodiles were apparently transported to Paya Indah from Langkawi. The rangers also state that they only feed the crocodiles 2 days on the weekend. Days other than that, the crocodiles will find their own food. Crocodiles are carnivores, so they only eat meat. And they often kill their prey by drowning them under the water, so it is easier for them to eat their prey. Pretty gory I'd say, but well, that's their nature. So yeah, that's all from me and I'm gonna pass the next part to Mira.
After we are feeding the crocodile, we walk for about 2-3 minutes and arrive at one small stall in that area. Because we were thirsty, we bought a beverage and various snacks to fill our bellies. After that stop, the rangers asked us to go to the next station, so we continue our journey to the next station. We need to walk about 2 kilometers to arrive at that place. So here is the place. We need to divide into groups and each group consists of class members to do CSR activity. We must plant about 4 to 5 plants per group. In this station, we learned about how to plant trees in the right way. So in the group, we started to plant our tree. We plant about 2 species of tree which are called Avoha Belimbi and Citrus Hystrix. And here is the montage.
Adalah hasil tanaman dari kami.
We went back to their hall and moved on to the lunch. And finally, Paya Indah Wetland served us with their appetizing food. And after we got the food, we were having our lunch together before we performed Zuhur. So here is the montage.
Next, we will design the second destination, which is Taman Botani Putrajaya.
We both completed the task of measuring and recording the length and width of the leaves alternately for this activity, because there are 3 sets of leaves which is equivalent to 30 pieces of leaf to be measured.
Question 1. Discuss the differences or similarity of the mean of length between different set of leaves. Do the same for the mean of width. Based on the experiment we conducted, we have obtained 2 types of values that refer to the length and width of leaves. There are differences in length and width between these 3 different sets of leaves. However, there is only a small gap between their means. Using the value we got, we calculated the mean, variance and standard deviation. A graph here shows the correlation between the length and width of the leaves, which is linear. However, the mean values for length and width obtained for each set differ. Sets 1, 2 and 3 have mean lengths of 18.38, 12.66 and 13.75 respectively, and mean width of 6.34, 4.67 and 4.02. These differences in value may be caused by errors during data collection or any other external factors such as nutrients gained by the leaves that result in different sizes of leaves. In order to improve the data obtained for future experiments, the size of the leaves chosen needs to be taken into account to avoid large differences in value.
Question 2. What is mean by variance? The leaf have different measurements of width and length. What is the meaning of this statement? Discuss with respect to variance. What is mean by standard deviation? The answer is a statistical assessment of the dispersion of values in a data collection is referred to as variance. Variance explicitly assess how distant each number in the set is from the mean and consequently from every other number in the set. The reason the leaf have different measurements of width and length is due to the use of different size leaf in our experiment. As we can see in the table here, the total value of length and width for each set plays a vital role in determining the mean value afterwards. Standard deviation can be defined as a measure of how much they vary or are dispersed. Additionally, it is used to demonstrate how measurements for a set of data are dispersed from the average or mean. The standard deviation is determined as the square root of the variance by calculating the departure of each data point from the mean. If the data points are further from the mean, there is a bigger deviation within the data set. Consequently, the higher the standard deviation, the more spread out the data. In addition, low standard deviation and high standard are the two categories of standard deviation. High standard deviation means that the values are dispersed over a greater range, whereas low standard deviation means that the values tend to be close to the set's mean. Question 3. Why is it necessary to have different sets of measurements for the measurement of leaf width and length? The answer is, the different sets of measurements for the measurement of leaf width and length help minimize any error during obtaining the data. Besides, we also want to see if the leaf share the same characteristics with each other or not.
After briefing, we start to walk around with our own group members to place that we choose for quadrat sampling. Our group choose the place nearby. Then we start to make quadrat. This is a short video during the process of making quadrat. The quadrat size is 10 × 5 since we want to explore more species. When we finish, we start to figure out the name of species in quadrat and record it. Next, we measure all the diameters of breast, height, and record it. Then, we gather all the information that we obtained together. Lastly, this is the result of our quadrat.
This table shows the species that we found in the quadrat.
We have posed that question. For the first question is define term quadrat in practice. In ecology, a quadrat is a frame that is typically square and is used to isolate a standard unit of space for the study of an item's distribution over a vast region. The quadrat is useful for collecting samples of sampling plant. Next is question 2. What are the two basic requirements of all the quadrat techniques? The area of the quadrat being counted is known, and the population in the quadrat area must be determined precisely. Next, for question 3, what would be the advantage and disadvantage of using a larger or smaller quadrat to survey the inventory organism? The advantage of using a larger quadrat is relatively simple to use, while the disadvantage of using larger quadrat is enlarging a sampling quadrat may overlap parts of the range of certain species that were previously absent and increase the estimated species density. The advantage of using a smaller quadrat is the species in the quadrat are easier to count, while the disadvantage of using a smaller quadrat is the quadrat is too small and may result in the emission of too many individuals, resulting in a less representative population size estimate. Next, as for the question 4 is, problem with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested area or in aquatic habitats where one cannot directly see the animals or plant of interest. Explain the issue concern ecologists in conducting this sampling method. The answer is, it might be challenging to use spatial sampling to analyze very heterogeneous regions. Heterogeneous refers to a research area where contents vary at different points across the area. If a section of the area that is different from the rest of the area is overlooked, conclusions about the entire region obtained from the spatial sample will not be completely right. Lastly is question 5. Why is there a difference in the number of plant species in the quadrant? Hypothesize or make your own assumption. Discuss the factor that would have caused this type of distribution. The answer is, according to my assumption, from the difference in the number of plant species in a quadrant depends on how well those plant species have adapted to that quadrant area. The number of plant species will be higher than the other species if the species is highly adapted to the environment. A species' ability to survive and reproduce, and consequently its local density and geographic distribution may be restricted by both physical and biotic factors. The consequences such physical factors on these species include rainfall and temperature. Abiotic factors such wind intensity and direction will increase the number of species in an area if the temperature is low, and the species are very durable to low temperature. Where organisms are found throughout ecosystem is greatly impacted by the wind strength and direction. Many species prefer more protected environment. Animals that depend on plant seed to reproduce are more likely to migrate and germinate there, increasing the chances hence they will live nearby. The growth of specific organisms can also be impacted by the winds